Good morning, everyone. In today's session, we'll discuss about IGCSE chapter 16. Sorry, it's chapter 15 as per the syllabus. The name of the topic is drugs, and this is very important for exam point of view also because they ask questions from this particular topic. At least one multiple choice question you will get from this topic and one 2 marks or 4 mark question in the paper 4 or paper 3. So let's go ahead with the topic. This topic is quite small, so we'll discuss basics about the drug, like what is drug and how you will define it. So the definition is given here that any substance taken into the body that modified. When we say modify, the meaning is it's going to change the rate of the reaction in the body or affects chemical reaction in the body. That's called drug. So if the question was define or describe, you can write this particular statement. Then we'll see describe the use of antibiotics for the treatment of bacterial infection. So we'll see why antibiotics are used for only for bacterial infection. That we will try to understand. Describe that some bacteria are resistant. So if when we are unwillingly we are using tablets whenever we have any fever or any mild problem, we take antibiotic and knowingly, unknowingly, we are making them more resistant. So that we have to avoid. Then we'll talk about state that antibiotics kill bacteria but do not affect virus. We'll try to understand this also, why. And the last we'll discuss about that explain how using antibiotic only when it essentially can limit the development of resistant bacteria such as MRSA. So we'll talk about one example also, how this bacteria becomes resistant. So without wasting time, let's go ahead with the topic and let's try to understand. So we are going to discuss drug. This is chapter 15 in current Cambridge syllabus. So first, Cambridge asked the definition or sometime they asked to describe. So when it comes to define, you can write one line statement, any substance that taken into the body. Right, so this will be 1 mark. Suppose the question is of 2 mark and you have to write the definition or you have to describe the term drug. So in the 2 marks, what all points will be covered? 1 mark will be given for any substance taken into the body. And the second marking point is either it modifies or affect the chemical reaction in the body. So this definition I have taken exactly from the syllabus point of view, and there is no changes in the definition. If you have written this definition in the exam, you will get full marks on this. Right, so next, so some drugs are medicinal drug, right? So some have medicinal effect like turmeric, neem, ginger. These have antibacterial property and they have medicinal purpose. Right, so some drugs are medicinal drugs that are used to treat symptoms or cause of a disease like antibiotics. So the liver is the primary site for drug metabolism. So whenever we take any medicine, your liver is the first organ in the body which controls, regulates, or you can say metabolize it in the liver. So liver is a checkpoint actually, where everything is being checked. Is it good or it is bad? If it is a bad, then liver will try to detoxify it. If it is good, then it is released into the blood. And then through blood, it will reach to the target part, and then it will affect there. Then after a few days, you will feel good. I mean, that drug has worked on the body. Okay, so this is basics idea about what is drug and how they work. Now, what is antibiotic? When we talk about antibiotic, first thing will come, it's a chemical substance. Always remember, it's a chemical substances. It's that made either from certain fungi or bacteria, right? And these antibiotic, what they do, they affect the working of bacterial cells. So now the question is how they affect, what they are doing. So they can do so many things. So it will either destruct, right? Either they will destruct, right? Disrupt what? They will disrupt the structure or function. Or second, what they will do? Prevent them from reproduction. So reproduction is also going to get stopped. Okay. So we know that these structure and functions and the reproduction is a property of a bacteria. So this will work best against bacteria. But not against virus, because virus is not a cell until unless it is not getting in a living cell. So before they don't have their own structure and function. Yes, the structure is there, but as they get entered inside the cell, they perform their own metabolic reaction by using all the cellular machinery of the cell. So antibiotic, what they do, they target the process and structure, processes and structure that are specific to bacteria. So bacteria are prokaryotic cells. So all prokaryotic cells will be affected by this particular antibiotic. And they generally do not harm the human body. So they are only going to affect the bacterial cell. Right? So like take an example. Suppose this is the cell. This is a penicillium. This is the drug. You can see this is the drug, right? Given to the cell. This is the prokaryotic cell, also called bacterial cell. So what they will do, what this drug will do? So drug will rupture the cell. So that all the cell content will come out. And second thing, prevent reproduction. This is again very important process. Now the question is how antibiotic works, right? So let's try to understand this whole paragraph. So I have given so many information here, but all are important as per the Cambridge exam. So let's start with the first. Since the first antibiotic was discovered in 1928, right? Then many more have been discovered and developed, and antibiotics were and after widely overused. So it's a continuous process. Every year, many hundred thousands of antibiotics are found regularly. Commonly prescribed antibiotics are becoming less effective. Now there is a big question why they are less effective due to a number of reasons. So now what is the reason? So number one, overuse. You're overusing it, even it is not necessary. So what is happening? The bacteria are getting resistant. Second, sometimes when you take medicine, you feel good after 2-3 days, and you stop taking the full course that is prescribed by the doctor. Suppose doctor has given 5 days of dose, and you have completed only three. Suppose you have done only 3 doses, right? Only 3 days, but it was prescribed for 5 days. You have taken only 3 days, so obviously bacteria will get resistant. And the third is, which is very important, Large scale use of antibiotics in farming. So farmers are putting antibiotics to kill the bacteria, keep it close even the animal is not actually sick. So which is not good. You are knowingly, unknowingly, you are generating a resistant bacteria. So what will happen? This has led to the effectiveness of the antibiotic being reduced. So which is not good. So if the effectiveness of the antibiotic is going to reduce, then the purpose has gone. Now they are not going to affect any metabolic reaction because bacteria are more resistant. So these bacteria are commonly known as superbugs, and the most common is MRSA. So bacteria, they are not going to get affected uh, by the other things because by the other antibiotic because they are totally resistant. They become totally resistant towards that particular drug. So there is no effect. So the most common type of superbug is your MRSA, also called methicillin resistant Staphylococcus aureus. Right, what they do, we individually can help prevent the incident of antibiotic resistant increasing include. So what are the reason behind it? First, only taking antibiotic when absolutely needed. So avoid unnecessary intake of drugs. And when prescribe a course of antibiotic, ensure that you must complete the course, even if you feel better after a few days. So that is very important. So in this diagram, we'll try to understand how they become drug resistant. So let's see number one, the steps. A population of bacteria in the gut. So gut is the small intestine. Some have antibiotic resistant property. So you can see here this purple color. They are pathogenic antibiotic uh, resistant bacteria. They are highly resistant. The red one is the pathogenic bacteria which cause disease, and the green one is the healthy one. So when exposed to antibiotic, bacteria causing illness, as well as the healthy gut bacteria also get killed. Right. So now you can see who is alive here. The purple one, which were resistant. With reduced competition for nutrition, antibiotic resistant bacteria multiply faster, right? And they will become more in number. There is a possibility that the plasmid with antibiotic resistant gene can be shared between the bacteria and both will do the same kind of property, like they will become pathogenic. So the method by which the genetic material is transferred from one bacteria to another bacteria is called transformation, right? So by transformation method, the genetic material transferred and the non-virulent bacteria can also act as a virulent bacteria. So that's it. That's all about the drug. We'll see you in the next topic. Till now, take care.